Okay, good morning, everyone. So I'll just share my screen. So this is my presentation, and yeah. So my topic was to perform the molecular docking of APP with Donepezil. So the key objectives of this presentation and this experiment were to perform the molecular docking of the protein assigned, which was APP, amyloid precursor proteins, and with the ligand, which ligand of choice. And I chose Donepezil, which is a clinically proven drug for the treatment of this disease. After that, I determined the binding affinity of I determined the binding affinity by analyzing the docking scores, and I also created 2D and 3D interaction maps with 2D and 3D interaction maps, and then I analyzed the results. So, coming to the introduction, so we all know that Alzheimer's is a neurodegenerative disease, and it is characterized by a cognitive decline in memory. Now there are various proteins involved in this particular disease, but and APP is one of them. It plays a role by forming of the amyloid plaque, beta plaques. Now I have chosen Donepezil as it is a clinically proven drug for the treatment of this disease. Moving ahead, these are the protein details. So the protein assigned to me was amyloid precursor protein, and I have taken my source as UniProt. I have downloaded the RCSB PDB file, and as you can see, this is the structure of the protein in BioVia. This is not a prepared protein, it's an unclean protein. It consists of water molecules, nucleic acid, and all the chains, and devoid of polar hydrogen. Coming to the ligand I chose was Donepezil, and I took it from PubChem. Now, the mechanism of Donepezil is the inhibition of cholinesterase. Now, this is the PubChem website from which I have chosen, and this is the ligand in BioVia. It is also not prepared; it is just visualized. Now, coming to the methodology, now I used BioVia for the protein preparation. I removed water molecules, nucleic acid, and unwanted chains, and I added polar hydrogens into BioVia, and then I uploaded it into the PyRx. The ligand was prepared and uploaded into PyRx itself, so it was minimized and converted into PDBQT format. After that, the molecular docking was carried out by using Vina Wizard, AutoDock Vina, in the PyRx software. The docking scores were analyzed by the output table, and the one which had the lowest binding affinity was taken forward for further visualization processes. And then I had created the 2D and 3D maps in BioVia and visualized the structure in PyRx. Now, the first step was the ligand preparation in protein. So this is the prepared ligand in the BioVia, devoid of everything, and includes polar hydrogen. Then this is the ligand in PyRx. It has been minimized and converted into PDBQT format in the PyRx software itself. Coming to the docking results, after docking was performed, this is how the structure looked like. All the poses were superimposed on each other in the same protein molecule. So for this, and these are the docking results. So there were 9 poses, and this, the one with the lowest binding affinity was the first one, which has a minus 7.6 affinity. So I went ahead with this one for all of my further visualizations. So coming to the then poses differentiation, I did it using the Vina split option, and I went ahead with the first one for further visualization. Now, coming to the visualization, so this is the output result in BioVia, and this one is in PyMol. So as you can see that this is the protein, and this is the ligand, which is fitted right inside the protein pocket. So this is the 2D interaction map. So this map shows the interactions of the APP at the specific amino acids with the ligand, and it shows the hydrogen bonding, pi stacking, as well as electrostatic interaction, which help to explain that how the ligand is anchored well within the pockets of the um, binding site of the protein. So this is the 3D interaction map. So this one shows how well the ligand has been placed within the binding sites of the protein as well. So it supports the stability of the dot complex. Both of these have been formed in BioVia itself. So this is the PLIP analysis. So I tried to upload the protein ligand interaction in PDB format numerous times, but this is what it kept on showing. Moving on, so this was the end of my project, and in conclusion, I found out that donepezil and protein have a great interaction, and that this drug can be used for the treatment of Alzheimer's as it is being used. But in silico, I found out that the docking score of -7.6 indicates a stable and energetically favorable interaction. And thank you. I also would like to mention that the laser pointer and all the other things were included because I recorded the presentation in the PowerPoint as well, and then I saw the guidelines. So that is why it was moving at its own pace, because I recorded it earlier in the PowerPoint presentation. So that is why. So I'll just stop the screen share. And yes, thank you so much.